I would like to welcome Jennifer Berry. For over 21 years, Jennifer Berry has been the apicultural research professional and the lab manager for the University of Georgia's honeybee program. Her research and objectives have focused on queen breeding, improving honeybee health, IPM techniques for varroa, and small hive beetle control. Also, sublethal effects of pesticides on beneficial insects, weeds for bees, and what is best to plant in a non-traditional horticultural landscape to enhance pollinator populations and diversity. Jennifer's extensive duties include teaching beekeeping to folks all over the world, those included incarcerated in Georgia's medium and maximum security prison. She is not only a PhD student, but also the instructor for the new Bees, Beekeeping, and Pollinator Conservation course at the University of Georgia this fall.
I would like to discuss with you a project that we've been doing for the last 3 years on oxalic acid. And before I get any further, I do want to thank the Georgia Beekeeping Association. They have been outstanding supporting our research at the lab, at the Bee Lab, and especially this oxalic acid work. And so without them, we would not be doing this. So I thank them for being so supportive. And I'm also going to just make a little mention. I have 7 animals in this house, and you never know when one's going to bark, or a kitten's going to jump on a lap, or knock over the computer, or dislodge a hairball on the table here, which happened a couple of weeks ago. Anyway, so let's get started. Why are our bees dying? What is the number one reason our bees are dying? Well, it's varroa destructor. It's plain and simple. There are other things, of course, pesticides, habitat deterioration, nutrition, starvation, but the number one reason our bees are dying is because of varroa destructor. Once varroa jumped from Apis cerana, the Asian honeybee, to Apis mellifera, the Western honeybee, it changed bees and beekeepers' lives forever. The problem was our bee, our European bee, the bee we have here now in the United States, has not evolved with this mite. And so Apis mellifera never developed any kind of resistance. Whereas Apis cerana, if you can find mites in Apis cerana colonies, they're very benign. They're there, but they have evolved over time to reach this kind of host pathogen equilibrium, so the mite doesn't do damage, or the kind of damage that we see in Apis mellifera colonies. So like I mentioned, Apis cerana and varroa evolved together, where Apis mellifera has not evolved for millions of years with these pests. And that's the problem. They don't have any way to deal with them. Now, there are a lot of viruses that are found in honeybee colonies, most of which are harmless or benign until varroa comes into the system. Once varroa comes in and begins to feed, that is when the damage occurs. Now, I'm just going to go through this real quick. This is varroa reproduction. We have the foundress mite, and that foundress mite is going to enter a cell. Once she enters that cell, she's going to nestle underneath the larva, and she's going to begin to feed. And then when that larva reaches out in that pre-pupa position and that cell is capped, what happens is varroa will begin, that foundress mite will begin to open up a port or a hole for her upcoming developing progeny. Then she's going to lay an egg, and that first egg is going to be a male. And then subsequent eggs will be females, and those females are going to mate back to the male mite. They're going to go through their developmental processes, and then they're eventually going to reach adulthood. Hopefully for us, after the bee has already emerged, but most likely they have reached adulthood when that bee is emerged. Let me clarify that. So If this bee emerges before their outer coat has sclerotized or turned a brownish color, has hardened, they will die. So they have to reach that adulthood. Otherwise, they will die when that bee emerges. All right, so varroa feeding on the developing brood, that is where the real damage occurs. Yes, varroa does feed on the adult bees, but this is where we start seeing deformed wing. And this is why we see deformed wing. Like I mentioned earlier, deformed wing has always been present in colonies, but we didn't see symptoms until varroa begins to feed. And once varroa begins to feed on that developing brood, those viruses get turned on, and then the bees become sick. In the good old days, most of the diseases that were coming in through the mouth, like our foul broods and our chalk broods. They were being introduced through the mouth parts. So the nurse bees, when they were feeding the developing larva, it's almost like mother's milk, where they're developing these immunities to a certain degree in the midgut. So when things would come in, they might not make it. Well, that's not the case now with varroa, because varroa bypasses all of that by directly feeding on the developing broods, not going through the mouth anymore. And what they're doing is they're feeding on fat bodies. And I'm sure a lot of you have heard about the research by Dr. Samuel Ramsey, who discovered that that varroa wasn't actually vampires and feeding on blood, the hemolymph of bees, but they were actual werewolves, where they're chewing and digging into and cutting into the fat bodies, which are rich in food. That's why the mites are feeding there. But the thing is, it's a very important organ. It's vital to the health of honeybees. It's kind of, we were to compare the human, it'd be like our liver, because where we store fat. And what do we need that fat for? We need that fat for energy. And so as the mite is feeding on that developing bee, it is it's causing the bee to become weakened, become sick, and therefore we start seeing the symptoms of viruses like deformed wing. So right here, right there is when the damage begins, by weakening the bee, and these viruses become active. So we have this beautiful colony, and we're really happy because it's end of the season. We're going into the fall. It's full of bees. It's full of brood. But what's been happening is, as our bee population has been increasing through the spring and summer months, so have our mite populations inside of those colonies, because the mites need the brood. And so they're going to be following right behind that brood. As brood is being reproduced and developed, so are mites. And at some point, if the colonies begin to taper off in anticipation of winter, because they know they need to back off on population size, well, what can happen is the mite population can actually exceed the bee population right here. And this is not good. This is not a good thing especially going into winter, because we need our bees to be their healthiest going into winter. We need the winter bees to be healthy and strong and not full of virus. Otherwise, they are not going to survive. So inside of our colony, we don't know what's going on because we haven't checked them in a while. Here's our beautiful queen. But do you see this deformed wing here? Also, they're chewing out the brood. This could be infested brood with mites. And you see where some of the larva looks, they call it snot brood or PMS. And what's happening is the larvae are dying. This colony is sick. They're loaded with viruses. And one by one, bees are dying, 100 today, 1,000 tomorrow. And slowly, this colony begins to crash to a point where there's not enough bees to even remove the dead bees. And the colony crashes. I hate this photo. It's very sad. And then we open up our colonies for a midwinter inspection, and there are no bees. Or we open up our colonies in December just to check, or November to check, make sure they got good populations, everybody's ready to go into the worst part of winter, and we find no bees. And that's because they've been dying over days and weeks and months, because as the mite populations are increasing, the virus populations are increasing, and our bee population is decreasing. A lot of times I will get calls this time of year I get calls all the time because that's part of my job at the university is extension. And I'll get calls saying, hey, I had this beautiful colony. It looked great. It was full of honey, full of bees. And I went in and looked at it and I found absolutely no bees. Or I found a tiny little cluster with a queen and no brood and lots of honey. What's going on? And my first question always is, did you do anything for mites? Or what did you do for mites? Have you treated for mites? And sometimes the beekeeper will say, no, because my colonies don't have mites. And I'm like, how do you know your colonies don't have mites? Did you do an alcohol wash or a sticky screen? Oh, no, I never saw mites. So my colonies therefore don't have mites. I'm like, well, you're some of the few colonies in the world, other than Australia, that don't have mites, but let's talk about it. And they say that they did not see mites. Well, do you see the mite here on this bee? And I know sometimes we're not paying attention, are we? We're not paying attention to looking for mites because we're looking for the queen or the pattern or how much honey, or is there pollen coming in, or we're just sitting there watching how wonderful bees are and they're emerging and they're doing dances and all that, and we're not looking for mites. Well, they're there, but they're on the underside because that's where the fat bodies are. It's on the underneath of the bee. So we would have to pick them up and look. And again, I ask you, do you see the mite here on this bee? It's hard to see. It's hard to see even for someone who's trained to look for mites. So about 90% of the time, the mites are hidden from view. They're either on the underside of the bee or they're in the cell under that protective cap. Well, how do mites become established in a colony? Well, one way is we buy them. When we buy nukes or packages, they're going to have mites, more than likely. And all it takes is one mite. Remember, that female mite, when she emerges from that cell, she has already mated, she's ready to start laying eggs, and she's ready to go into a cell. So it just takes one mite. If you buy that nuke and it has one mite, your colonies will have mites eventually, and a lot of them if you do nothing. Same with packages. Also, too, bees drift. Bees drift from colony to colony, especially drones. Drones are notorious for drifting, and they're going to drift with mites. And as they go from colony to colony, a colony that may not have a lot of mites, all of a sudden can be overtaken by mites, or all of a sudden where you do an alcohol wash and you found there were no mites, and then a month later you do an alcohol wash and you find you've got 5 mites per 100 bees. So it can happen very quickly through drift. Also, if colonies are crashing around in the area and bees are going out and robbing colonies that are crashing, what are they doing? They're bringing home those mites and some honey too, but they're bringing home mites as well. So what can we do to save our bees, and how many mites are too many? I apologize for this slide, but it's so crucial. And I do it in my own operation at the lab and in my own personal beekeeping operation. I do integrated pest management. And this graph right here is kind of the key to integrated pest management. I've been talking about it for 20 years. I know some of you have probably heard me talk about it and are tired of hearing me talk about it, but it is so important for us to understand. So this is really going out to the new beginners, the new beekeepers out there. You need to know when a pest has reached a population where they are going to cause damage. And in this particular graph, we have number of pests, and they could be anything. It could be tomato hornworms on your tomato plants. It could be aphids or whiteflies or fall armyworms in a pasture. And then it's just over time. And what this red line is indicating, it's showing this pest population is starting off very low, which it normally does, and then it gets higher and higher because there's nothing here that is controlling that pest. Say it's an invasive species, or it's like varroa or small hive beetles. Nothing is controlling their population, their reproduction growth. So they explode, and very quickly they can reach the economic threshold. And that is the number of mites, of aphids, of tomato hornworms, of pests that we as the beekeeper, the farmer, the gardener, whoever, needs to do something. Because if we don't do something at this point, what happens is those numbers continue to climb, that pest population continues to climb, reaching the economic injury level. And now we have lost our colony, or we have lost our field of hay, or we've lost our crop of tomatoes because we didn't do something at this point where we should be controlling, and that population was allowed to exceed. And the reason they say economic is because just like us as beekeepers, we've got a lot of money invested in these colonies, in these bees, especially on a commercial level. And so at some point, the mites are going to pull those numbers down, are going to pull whatever, our honey production, our nuke production, to a point where we've got more input, we have more money in than we're going to get out, because the mites are causing an economic decrease or decline. And so we call that the economic injury level, because if we lose that colony, we just not only lost the money we put into it, but maybe even our business, if we are involved in a commercial operation. So right here, okay, this springtime, we need to be out there checking our colonies, what's going on, what are the populations like, and doing all of our colony assessments. But really important, what are our mite loads? What's our mite population at that point? We can do it with sticky screens, which is non-invasive. You can buy these through Daydant. You stick them to a board with a screen, and you insert them into the colony, and you leave them in there. We leave them in for 3 days. And then we pull them out and we count the number of mites on the entire screen. And then we divide that number by 3 to give us the 24-hour mite drop. All right. Now there's also powdered sugar, where you can take 100 bees and roll them in powdered sugar and count the number of mites. I do not recommend this one. Yes, it does not kill the bees. It still harms them. It doesn't kill them, but it's not a real accurate assessment of the number of mites that you have in that colony. What I would recommend, and what we do at the lab in conjunction with sticky boards or sticky screens, is we use an alcohol wash. Yes, it does kill the bees, and I hate it, but I need to know what are our mite loads in these colonies. And if I need to know it very quickly, what we do is we fill this with alcohol. We have a scoop that accurately counts about 100 bees. We drop it into the little container and we shake it, and the bees die, but so do the mites. And the mites fall off and they float to the bottom, and then we can count the number of mites in the colony. And that gives us a % mite load. If there's one mite per 100 bees, 2 mites per 100 bees, 10 mites per 100 bees, whatever, that gives us a %. And then we know we can take these numbers and figure out what we need to do as the beekeeper. So for sticky screens for a 24-hour natural drop, we're looking at about 12 mites. And I keep saying I'm going to change this slide. Don't look at 38, just look at 12, okay? 12 mites, powdered sugar roll, 1 is too many. And an alcohol wash, 1 or even 2 is too many. Now it's also kind of depending on the time of year, spring versus fall, but we need to know and monitor our colonies to see what are our mite populations. Now, for years, I've been saying, once you find out, you do your alcohol wash, say you have 10 colonies, you do 10, and you find that only 5 of them have 1 mite, the other 5 have 0 mites in 100 bees. Well, we would have said, just treat those 5 that have reached the economic threshold. And this was really important back in the day when we had compounds like coumaphos or fluvalinate, where resistance was building very quickly. Nowadays, we have compounds like Apalife Var and Apagard, our thymol-based colony, or thymol-based miticide, sorry, that we don't think we're going to see much resistance just because of the mode of action there. So what I recommend now, and I do personally, is we go in and we check a couple colonies, and if we've reached the threshold, I'm going to treat the entire apiary because of what we have seen in research that we have done and how quickly mites move and how quickly they're drifting from colony to colony. So look at the apiary as the superorganism. Look at the apiary as one colony. And even if you have 30 colonies in there, that apiary is going to be moving and it's going to be constantly in flux and changing, and those mite levels are going to constantly be changing. So, all right, let's have a little fun for a minute. So I did a Google search, and my Google search, I asked for how to control varroa. And these 10 things popped up, and a couple of them are actually pretty good, but there's this one here, controlling hives naturally with food grade mineral or with mineral oil. This one is using moonshine and oxalic acid. This one's using wintergreen. This one's using drone brood removal. This is using lavender. This is a homemade version of formic acid. This was some weird screen I don't know. I couldn't understand. And then this is oxalic acid. All right. So out of, let's say the 9, because this is actually just a little video, out of the 9, how many are legal? Well, actually, only one, this oxalic acid, that's legal. Formic acid is legal, but not making it on your own. Lavender, even lavender is technically illegal. You're not supposed to put anything in a colony that does not have a label. And that label is so important because it's going to protect your bees, because it's going to tell you how to apply it. It's going to protect you because it's going to tell you what you need to wear or not wear. And it's also protecting the environment. Now, granted, this drone brood removal isn't illegal, but here's my other question. Has it been tested and does it really work? I know that Mr. Don, the Fat Bee Man, has a huge following on YouTube. And he has people coming from all over and pay him hundreds of dollars to work his bees. And he's always on talking about this or this new thing or what he believes, but has it been tested? I know sucroside and some other things, mineral oils, have been looked at and they weren't even worth continuing because they do not work. Now, so let's go back to Google. And again, we're going to have a little fun here. We're going to go back to Google, and I did a search, how to control varroa, and I found that fairy dust works. I said, awesome, this is great. So I ordered the fairy dust, and because I'm Amazon Prime, I got free shipping. It showed up in 2 days, which is crazy. And I went out into the apiary and I took the fairy dust and I sprinkled it on a colony, and guess what? That colony survived. It was wonderful. So I'm like, man, this fairy dust is pretty good. However, but does it really work? So what I'm going to do, I am going to take it into another apiary and I'm going to apply it to one colony, but I'm not going to apply it to the other colony. And I'm going to see whether or not it works. Well, guess what? The colony I applied this fairy dust to survived, and the one I didn't, it died. So it's got to mean that the fairy dust is working. So now I'm going to go on YouTube and I'm going to start telling everybody they got to be using fairy dust on their colonies because I tried it. My colony survived. The one that I didn't try, it died. Now, does the fairy dust really work? Okay, I know I'm being real simplistic here, but how many times have we gone on YouTube or on the internet and read something and believed it and didn't even, I do it all the time. I'll look up something. Wait a minute. Is that even true? So just because it's on the internet doesn't mean that it is true or that it's going to work. Because my question about this fairy dust and all these other things that are on the internet right now that supposedly are working, is it really working? Did they collect any data prior to putting in the fairy dust or whatever material during and after? Did they check mite loads before, during, and after? What about the population of bees and brood? What about the health of the colonies? Or was the colony even queen right or queenless? Did they swarm? What about the location? Was it there in different locations? And the most important thing, can I replicate this over and over again and get the same results? All right, so why am I telling you this? So when we ask a question, does oxalic acid work? We do an extensive background research to look at all the other studies that have looked at oxalic acid or who not. Like if it was fairy dust, how many labs have looked at fairy dust? Once we realize or we find out that no one has really done a good background or done a good study on fairy dust, then we can construct our hypothesis. Will it work? Will it not work? Then we're going to take it out into the field and we're going to test it. If we are finding that it's working, we're going to analyze our data or find out that the procedure is working, not necessarily if it's killing mites or not, but the study is working. Then we're going to collect our data and we're going to analyze it, and then we're going to communicate our results. When we do studies, we need to do a minimum of 10 units, 10 colonies per treatment group, or 10 units. And then we need to replicate those units across different locations. Very, very important, because there could be apiary effect. Even if they're close to one another, one could be by a river, or one could be by a commercial beekeeper, or whatever. So we need to replicate across different locations, and this takes years. And that's the problem with so much of this research, is we can't just go out and sprinkle our fairy dust and say it works. We have to keep doing it over and over and over the years, different apiaries, different locations, different universities, Auburn University or USDA or Beltsville or where, so that we can construct something that is going to have reliable information at the end of the day. I know most of y'all have heard of Randy Oliver. He's a prolific writer for American Bee Journal. I don't think the man sleeps, because he's constantly putting up stuff for beekeeping and beekeepers. He's one of probably the most passionate people that I know when it comes to the bee arena. And years ago, he read about this material, oxalic acid, being mixed with food grade glycerin and putting it on a shop towel, and it was working against mites in Argentina. So he decided to try it in California, and he took the oxalic with the vegetable glycerin. He mixed it with sugar syrup, and he soaked shop towels, and then he put them on his colonies. And he was finding that they were working. It was killing mites. He wrote about it, and on the minute I read about it, I called him. I said, Randy, you're California, we're Georgia. You're hot and dry, we're hot and humid. Let's try it here. And so I got together, the reason too, Randy was saying that mites were dying, and that because of this process, he was able to keep mite populations down. So we agreed we were going to do it here in the Southeast. And my questions, is it going to work? If it does work, how well is it going to work? If it just works 10%, 50%, 60%, that's not good enough. We need something that we're going to put in there that's going to give us good mite drop, and not something just on the fringe. And then, is it going to kill or harm the bees, the brood or queens, or hurt us? And will it contaminate the honey? So these were our questions. We formulated our study. A lot of people got involved. I asked Dr. Jeff Williams over at Auburn University, and so he brought in his crew. We had our crew from the Bee Lab. Bob Benny, Blue Ridge Honey Company, volunteered 200 colonies along with his crew. Dreamhaven Farm, Sherrod Turton, volunteered 200 colonies in his crew, and we went to work. And for over a year, we spent time in 400 colonies, multiple locations. We had 4 treatment groups, a control shop towel with just vegetable glycerin, shop towels with 12 grams of oxalic acid, and then 18 grams. Here's our crew. This is while we're in South Georgia. And before we could start, before we even applied the first shop towel, we needed to have colony weights before, during, and at the end. And we also needed to know how many bees, how much brood, and really important, how many mites are in each of these colonies before, during, and after the study, because we need to measure, is the oxalic acid detrimental to brood? Well, we'll see, because we know what the beginning, how much brood there was in the beginning, and then we're going to do it at the end, in the middle. So we would be able to tell that it's detrimental to the brood. And here's just pictures of us taking a lot of data in the field. Now we're in North Georgia. It was much cooler up there. That's why we're smiling up in North Georgia. But a lot of time, a lot of people, a lot of work, a lot of shop towels, and a lot of mites were counted during this study. Well, we were encouraged because we were seeing mites dead on the shop towels. And the thing that's so nice about this, you put it in there, you're going to leave it in there for 21 days. You can come back and put a second application, leave it in for 21 days, and we're done. That's the beauty to the whole idea of this particular method of mite control. And Randy was saying, hey, folks, it's working. I'm getting great mite control. And he was writing about it. We collected all of our data, and here is what we got after 42 days, 2 applications. We absolutely saw no difference between colonies that did not receive shop towels and colonies that had 18 grams of oxalic acid. No difference, unfortunately. We decided to trial it again, and we extended the period of time, adding again a second application. And the orange is oxalic acid treated colonies. The blue are our control. And as you can see in some of our apiaries, we actually had more mites in the ones that had the shop towels than the ones that didn't. I know work still needs to be done. Randy is still using it, or he's moved into a sponge. We have not yet gone there because we wanted to trial some other things, which I'm going to talk about here in a second. But as of right now, our results show that we did not have any control whatsoever using this particular method. And also EPA is not happy about it because you're mixing oxalic acid, which can be very dangerous, on your stove and soaking shop towels. So this method is not going to ever get approved, at least right now, because there's no efficacy. We're not seeing any kind of control. And even if there was high efficacy, it would have to be packaged and in a safe manner so that people could use it. Sorry. So let's talk about, there are 2 approved methods for oxalic acid for varroa control. There's the trickle, which I won't talk about, and then there's vaporization. And vaporization, you can buy the vaporizers, and you can buy the oxalic acid. I highly recommend that you buy it from a reputable source, and the one that has a label that is for honeybee colonies, because it's going to tell you how to use it, how to mix it, how to apply it, how to be safe. And I would recommend going the legal route when you're purchasing oxalic acid. And then here's the wand. We get all of our stuff from OxyVap through the dealership there. And what you do is you put the crystals inside the little heating plate, and you connect it to a battery. It heats up and it forms a gas. Oh, and before you do that, you need to be wearing a respirator. Extremely important, because this stuff, it is dangerous. You do not want to breathe it. And then it creates this vapor and it penetrates the colony. Well, you might be doing this, or you might have seen this or heard about this, but a lot of beekeepers are doing the oxalic acid sublimation multiple times, 7 times, 5 times, 4 times, 3 times, so many days apart. And so I decided, let's do it to the extreme. We're going to do it 7 times, 7 applications of oxalic acid, 1 gram per super. And we are going to do it 5 days apart. Okay. And we did this over summer, and here are our results over multiple locations, a lot of colonies, absolutely no difference between colonies that we oxaliced and colonies that did not receive oxalic. Now we did this midsummer where mite loads were reaching the economic thresholds or had gone above the economic threshold. Would this kind of treatment work early in the spring? Because I don't know. We did it in a time where the mite loads were heavy and we got no control whatsoever. I have beekeepers who tell me they swear by this. They use it all the time. It's working for them. I can't answer that. I don't know why. But in our 100+ colonies, it did not work at all. So we've known this, and it has been told to us over and over again, you've got to have broodless colonies in order for this method to work, because the oxalic acid does not penetrate that wax capping. So all those mites that are inside of that cell reproducing are not even going to be touched or harmed by oxalic acid. So what we need to do is create broodless times. You up in Ohio during the winter months, perfect time to treat. Hit them when all the mites are going to be phoretic and running around inside of that cluster. That is the perfect time for you to treat. But let's say now we need to do a summer treatment. Well, what we can do is, this is actually our study. We had 90 colonies and we had 3 applications of oxalic acid 5 days apart. We had 6 treatments with a brood break and with no brood break. So we had our 0 gram, which basically is our control. And then we upped it up to 2 grams. I think we're going to get approval for 2 grams per super, because the 1 gram is just not strong enough. And with Dr. Cameron Jack's work down in Florida, he was showing that 2 grams is beneficial, whereas 1 gram is not. So we decided to test with 2 grams and then again with 3 grams, just to see, could we get even better control, especially in this group that did not have the brood break. Were we able to get some control? Here's just some photos of us out in the field. Here we are doing our colony assessments, our before, during, and after. How much brood, how much bees, how many mites. And this is Dan Harris. He volunteered his colonies for the cause. And Dan Long, here's Dan Long and his colonies. Very generous beekeepers here in the state and the world. Beekeepers are awesome. I just love them. But anyway, they've been so helpful, as we could not afford to buy hundreds of colonies, so we were able to use theirs. Anyway, one thing that our thought was, how would we create the brood break? Well, we thought we would cage the queen. That's what the Italians have been doing for years. They cage the queen, and then they come back 21 days later and they oxalic. Well, how many of us want to pick up a queen and put her in a cage, even a bigger cage like this? I don't want to do that. So what we did is we basically sequestered her above in a honey super that was honey bound, and I gave her 1 frame to lay on. And so she was up there, and I only kept her up there instead of 21 days. We only needed to keep her up there for 14, because even if she starts laying on day 14 and we're treating on day 21, the majority, if not all, of the brood is still going to be uncapped. And so what we did is day 14, we removed the queen excluder. We did look for queen cells and we didn't find any. We found very few. And then on day 21, we came and vaporized 2 grams or 3 grams per super 3 times 5 days apart. I apologize that this is not pretty yet, because we just finished analyzing this data and it's not in a nice, pretty form. But what this one is right here, this is brood. With brood and with no vapor. And this is without brood with no vapor. This is with brood and that got vaporized. And this is without brood and got vaporized. Don't even pay attention to those other 3. This is the one we need to look at. We got tremendous control when we combined the brood break and the oxalic acid. Now, I have not yet analyzed between the 2 grams and the 3 grams to see if there's any difference there. Did we get even better control with 3 than we did with 2? That's still to be looked at. But what we did see is that we got control, and actually we got such control that we even dropped numbers down well below the economic threshold. And some of these colonies were loaded. So we were very excited. It is working. Now I need to tease out, like I said, between the 2 gram and the 3 gram, and hopefully we'll have some answers. And hopefully this will be impressed. I'm hoping to submit this this year, and hopefully go through review and be in press and release next year, but I'll still be talking about it before then. Also, so that's our studies. Something else that you can do if you buy a package, oxalic acid works great on packages because there is no brood yet. You can dump your colonies or dump your bees into your woodenware, and then, in a couple of days after they've settled down, come in the evening hours, and when everybody's home, and do the oxalic acid. And you can really do a number before there's any brood, before any of those mites have entered the brood cells. And this is something you can do in the spring when you get those packages. Bam, zap those mites, get rid of them as quickly as you can. And then what we do is we tweet. No, we don't tweet. We treat twice a year. We do it in the winter months and we kind of assume that we might have some broodlessness in between Christmas and New Year's. It kind of depends. I'm looking at temperatures. When we've had a real nice cold spell for a while, that's when I'll go out and usually treat with the oxalic acid. And another thing that we're doing and following up, and we're actually collecting data this winter, is we're going to do 2 applications of the oxalic acid over the winter months. And I'm doing this because of a friend of mine who's a commercial beekeeper. He relies solely on oxalic acid, and that's what he did the last 2 years. And we were going to use some of his colonies for a study, but there were no mites. And that was in July, and he had done the treatment in December. So we were quite excited about that. Please remember that the oxalic acid is not penetrating the cappings, even if you're doing multiple applications where you think where we think that, oh, we're going to capture all the mites. We're not, because they're not all emerging at the same time, and they're all not entering the cell at the same time. And so we have constant movement of mites in and out. So it's not going to work. And we showed that even 7 applications 5 days apart, we were not capturing the mite numbers that we needed. And real quick, this is another popular one on right now, where they're taking oxalic acid, mixing it with ethanol or moonshine, and putting in an insect fogger and fogging their bees, saying they're getting great control. Well, it's totally illegal. It's a waste of good moonshine. But more importantly, has it been tested? Is it really working? And just because a beekeeper says, yeah, it's working, what was the number of mites prior to them using this treatment? And what was the colony condition? These insect foggers are more expensive, or close to, if not more expensive, than the vaporizers that you can get, which are legal, have been tested, and are proven to work. All righty, so we know oxalic acid works, so please go with what we know, and careful of what you're using and what you're listening to. Just get it from reputable sources if you can. And a great place to start is your state organization and your local organizations. We have also tested an OA glycerin product from Argentina for possible US registration. We just wrapped that one up as well this summer, and we have not analyzed the data. So I really don't know. I can't tell you. When we do studies like this, we don't label colonies. These are treated and these aren't. We want to be as blind as possible as we're collecting data, so there's no bias, because I want oxalic acid to work, or I want the allium cap to work. But I can't be out there collecting data and knowing that. So we do everything blind. And anyway, I just don't have any conclusions whatsoever on this new product. I really do hope it works. Real quick, what is it? It's oxalic acid that's already been incorporated into a strip. You apply that strip, and then and you leave it in for 56 days, so it captures all your mites coming in and out of the cells. All righty. So let's wrap it up. How can we prevent our bees from dying? Keep them fat and sassy and keep those mite populations low. Keep the mite populations lows. I guess I needed to do a grammar check on that. But anyway, you get the gist. Keep them well fed. Make sure they've got lots of honey on them going into winter, and get those mites off of them now while we're still making those winter bees, because they cannot be compromised with viruses or they will die. Lots of folks to thank. Again, Georgia Beekeepers Association, the Auburn Bee Lab, Dr. Jeff Williams and his crew. They're awesome. Just love them to death. Blue Ridge Honey Company, Dan Harris, Dan Long, Lake County Beekeepers Association, and Dream Haven Farms. So many people have been instrumental in our work, and I just give big hugs to everybody. All right, and of course, we've got to thank our fairy because she did some hard work too. So, and thank you all. And thank you for listening. And please take care of each other and your bees. And I cannot believe that my little Mosley, this is Mosley and Clarence, they've been running around underneath, but they were very good boys and did not jump up here. So anyway, I'm going to end now. And I guess I'm hoping that I'll take some questions. Thank you guys. Have a great day.